Well, hello, everybody. My name is David Carr. I'm with the Finger Lakes Institute. I'll talk about that in a minute. Manage the Starry Stonewort Collaborative. Thrilled you're all here. Thank you for taking the time to listen to what I have to say. I got a lot to cover, but I think it's all important. We hope that at the end of this, some of you will be inspired to go out and do some rake tosses and surveys for Starry Stonewort and other macrophytes. I want to thank Carol Cole too. She was integral in getting this to happen. We've been doing these long distance, which, excuse me, which was part of the plan, COVID or not. But it's a little challenge trying to do training when you're hundreds of miles away from people. So with that, I'll jump right in here. We are recording this. Everything you'll see today, you will have access to, if you don't already. And I'll go from there. This, I'm not sure you've seen this or not yet. I tried to circulate this or give it Carol to circulate. If there's any one thing you write down today, write this down, or we'll email it to you or something. I set up if you ever use Box, it's an online, Box is an online storage area, in the cloud, so to speak, and they've put 10 different documents up there, plus this actual presentation, and there's other links in the documents to other things and so on. And I'll actually be showing you that later. The other thing, real quick, housekeeping, probably everybody mute themselves. So the fire engines that go by and dogs barking, who knows what, doesn't get crazy with all the sound, because we've got 14 people on. I'll also stop once or twice during the presentation for like 1 or 2 questions, and then we'll do a big thing questions at the end. Use the little chat box is ideal, because then you can, if you think of it during the presentation, you type in a little thing in the chat box, and Carol's going to try and keep track of those. And that way it's not too confusing. We've got 14 people on here. Quick agenda. I'm going to talk a little bit about the collaborative, the background, and where I'm from. We'll talk about invasive species in general. This might be repetitive for some, or redundant for some people. A lot of you are probably pretty well informed, but it's good to go over this stuff. Then we'll get into the good stuff. We'll talk about the survey and sampling steps that we are suggesting you get into, including using the Survey 123 app. It runs on a phone or a tablet. It's a way to record the information you gather out in the field. And then we'll also talk about identification, and this gets into hardcore stuff. We'll do the best we can with identification. It's tough long distance like this from slides and pictures, but it's what we have. And we'll do the best to get you through that. And then there'll be some wrap up. I'm gonna make you work. I've got a little Zoom poll of all of 4 questions. It shouldn't be too hard, but it just gives some feedback and we can talk about things. So, background of the collaborative. Whoops. Okay, as I said, I work for the Finger Lakes Institute. That's at a college called Hobart and William Smith College in Geneva, New York. Great organization. As it says there, we promote environmental research and education. And we do it in several different areas, research, education, economic development, outreach. We do a lot of field work. We have a couple small labs on the premises. It's just a really neat organization. And then housed within there is this long name, Finger Lakes Partnership for Regional Invasive Species Management, which is a New York State thing, but the reason I say it is when you look at some of the supporting documentation I provided, you may see this acronym FL-PRISM, just so you know what that is. And we partner, as the name would imply, with pretty much anybody that wants to help reduce the spread of invasive species, prevent them, control measures, all those good things. It's a neat program. There's actually 8 prisms across New York State. And we're, of course, in the Finger Lakes region where I'm from. So we are funded through the Great Lakes Restoration Initiative from the Environmental Protection Agency. Thank those folks. This is a Great Lakes basin-wide project. This is a big change and a big growth for us. It's a way for us to spread our wings a little bit. Up until now, we've stayed within our region in the Finger Lakes in New York State. But this is a neat program. We're pretty excited about it. I have a map coming up. And the whole reason for doing this is to help pretty much anybody from the experts to the managers, the stakeholders, citizen scientists to address problems with Starry Stonewort. And so we're looking at 3 basic areas: ecology, outreach, and control techniques. And I want to mention a little more about control techniques right now. That's on everybody's top of everybody's mind, of course. Of course, you can't do good control without the other 2. But there's tremendous research going on now with control techniques. One of the things we wanted to do at the onset of the collaborative was talk to the experts across the Great Lakes Basin, gather all the best management practices, disperse them, maybe come up with our own. And I actually physically, met in person with a bunch of the experts early on, and they looked at me like I was crazy because there's no known best management practices for controlling Starry Stonewort, which is that's the bad news. But we're working to change that. So there's tremendous work going on right now towards new and different control techniques, modifying them everywhere from chemical treatment to hand pulling to everything in between. And I'm confident at some point we'll have something that'll at least work on early beds and things really efficiently. Real quick, Great Lakes Basin, the big area, that's the actual watershed line or basin for the Great Lakes. International, talking to you folks in Canada, it's pretty exciting. We got, in a very short time, there's a whole bunch of folks up in your region. Some on this call, I've talked with and worked with Brian Ginn from the Lake Simcoe region. Just last week, I talked to folks towards the Napanee, on the east end of Lake Ontario from Nature Conservancy Ducks Unlimited. So I put a little star up roughly where you guys are from. I know you're from a lot of different associations. And then that little flag in the lower right is the Finger Lakes Institute. I'll zoom into that real quick. That's where we are, the Finger Lakes. It's a real pretty area. We're proud of it. The lakes are shaped like fingers, obviously. Long and skinny. The lake we're on there is Seneca. The one just to the east is Cayuga. And to give you an idea, those are both about 38 miles long. And then far over, you see my pointer here, over here is Auburn. I point that out because I actually live here. And this is a Wasco Lake. It's about 11 miles. And I actually work with the local lake association for this group too. But it's a beautiful area, lots of vineyards through here. You do wine tastings and things. I urge you to come in the summer. I know you folks know what winter's like too, but it's pretty in the winter too, but summer is the best season. Oh, and I wanted to point out too that that green line, it takes, that's part of the, can I go back here? One of the reasons we're so into it, this is like a third of New York State. And that's all part of the Great Lakes Basin, in our case, part Lake Erie and part Lake Ontario. So, we spread our wings, but we have a real stake in this too. It's all part of the Great Lakes Basin. So it's all important to everybody. This is just a little diagram of how we structured this thing. The Finger Lakes Institute's at the bottom. My desk is way down here at this little point. But we just we manage the program and facilitate it and so on. We have a group of 8 expert panelists, and these are folks that have been, in some cases, studied Starry Stonewort for 10 years, and they're from all across the Great Lakes Basin and beyond. Nice folks. They're all, high-level academics or state people, all researchers, and they act as like an advisory panel to me, basically. I talk to them on the phone. We have conference calls 2 or 3 times a year. I ask them questions about, should we do this? And they say yes or no. And so very helpful group. Collaborators are next. And these folks, I'm not belittling collaborators. Some of these folks have got as much experience as the expert panel. And these are folks from academics, state agencies, non-government organizations like the Nature Conservancy, just all kinds of people. And they act as we have like 30 plus people on our list for the collaborators now. And they're kind of our eyes and ears out there as far as what's going on in big projects, programs. They're also, we ask them to be our conduit to the most important piece, which is citizen scientists. And this is really working well. Early last week, I did a program for a group in Indiana, and that was the collaborator out there was at Grace College, the Lilly Center for Lakes and Streams, and they help organize citizen scientists, and we have a nice group there. That's exactly how this works. Now this group, I got to say, I'm going to promote not officially or officially promote Carol the collaborator, because without her, this wouldn't all happen. And so this is really exciting because we kind of skipped the collaborator stage here, and just a whole bunch of you folks are interested in what's happening. You're citizen scientists on the boards of lake associations and related, and this is a grassroots effort, and it's really exciting. I mean, this is what it's all about. But at the same time, we made some connections over the last, how long we've been talking, Carol, but 6 months to a year. And I know some of you folks have talked to the Lake Simcoe group, Brian out there, and I know you've talked to Amanda at the Nature Conservancy. So the connections were made, but it kind of came from the grassroots level, which is just really exciting. That's really neat. You folks are just concerned about what has to happen. So I'm going to jump right into some basics about invasive species. Again, this might be a little redundant for some of you, but it's not. It doesn't hurt to go over it again. Invasive species are non-native to an ecosystem, and it can often cause economic, environmental, or human health issues. Economics, impacts in ag and forests, and tourism is a big one we're looking at, probably for you too. When the Starry Stonewort clogs the boat channel, people stop using it, for fishing and swimming and all those things. Those are potential tourist dollars that are going away. Environmental, don't have to tell you probably, virtually any invasive species will mess things up. It'll push out native species. It'll break down natural processes and systems. And human health. There's direct and indirect problems with human health, and I got some really fun, scary ones to show you in a minute. This slide you may have seen, may not, but it's a really good slide. It's called the invasion curve. I honestly don't know where it came from. We show everybody this though, but briefly, you got time going from left to right, and then control costs going up as areas infested increase by invasive species. The lower left little section here, prevention. There's no species. This is where we want to be, and we want to keep them out of here, right? Then you get introduction of a species, and right in this early time, this is the best time for early intervention, okay? Intervention and early detection. I added this little early detection, because here there's a chance through this box of eradication of getting rid of it while it's still early on, whatever it is. In some cases, as you know, Starry Stonewort is taking over, but if you can find new infestations, there's a chance you might be able to get rid of it. And then containment, as we go up the ramp here, you got a lot more, bigger distribution of the invasive species. Eradication is unlikely, but maybe you can contain it. And unfortunately, this is where most public awareness typically begins. And we need, as a very large team, this is a community effort. We need to educate the public about down here. We want to keep it out, or if it's started, keep it from spreading. And then up in here, the bad news, this becomes virtually impossible to eradicate, becomes a management situation, and protecting, keeping things from spreading maybe, and it gets very expensive out this way. Now you're talking about running harvesters every year maybe, just to knock it down, maybe chemical treatment to knock it down. It all gets very expensive. And with Starry Stonewort, as I said, there's no quick fix at this point. Just a couple more things. Invasive species are sneaky. They have no natural enemies generally, high rate of reproduction. They adapt and to the very harsh conditions. As we said, they affect the biodiversity and ecosystem. They threaten native species. They outcompete with food and habitat. And this goofy cartoon, I just love this. We have a fair amount of milfoil and zebra mussels we've been dealing with out here for probably decades. But anyway, it's just a funny cartoon. Maybe not that funny, but okay, impacts. I just have some examples of a variety of invasive species here that aren't necessarily aquatic, but get the point across. Japanese barberry is ornamental. You often buy it at nurseries and greenhouses and stuff, and you often see it like this at commercial areas and things. It's a nice thing. Down here, this is what it looks like it gets loose in the woods in a forest. It takes over the complete understory, grows very rapidly. So folks that aren't familiar with invasive species, they go, well, so what? Well, I say, if you like to walk in the woods, some researchers looked at an area like this that's full of barberry, and they found as many as 120 ticks per acre. Without the Japanese barberry, they found maybe 10 ticks per acre. So an example of how, that's an effect on humans. I don't like ticks myself, so something to keep in mind. Giant hogweed. I hope you guys don't have this out where you are. We have it here, particularly up north in our county. It's an interesting looking plant. It's 6 to 8 feet tall and it grows very fast. It's aggressive, but it has a phytotoxin. I'm not sure that's the right term. If you touch anywhere on the plant and get any little bit of sap on you, it burns your skin. And I've heard it's worse, much worse than poison ivy. It's also photoreactive, so if the more sunlight you're in with the stuff on your skin, the worse the burn gets. I've heard it likened to chemical burns. It's just nasty stuff. Spotted lanternfly. This really cool looking bug, but it's coming up in our area again from New Jersey, Pennsylvania. They've got infestations down there. And New York State Department of Environmental Conservation, Department of Transportation, everybody's scrambling to keep this thing out of our state because it loves fruit. And we pride ourselves in our apple orchards, our apple crop, and as I said, our vineyards. We have a lot of wineries. It doesn't actually eat the fruit, but I guess it sucks the sap out of the tree or the plant and kills it. So it's a real problem trying to keep it out of here. Again, that's a major potential economic hit to our region if it gets here. And then just because we're here, Starry Stonewort, I don't have to tell you. Thank you, Carol, for this photo. I've actually used a lot of your photos, Carol. Dragging a boat through there's no fish habitat anymore, et cetera. It's just a big impact. So as I said, this is the thing right here to tell everybody you can and do it yourself. Preventing equals protection equals money saved. And even though if it's already established, you can keep it from spreading. That's what it's all about. We have a very active watercraft steward program down here. We put usually college students at any number of different boat launches and stuff, and they try and educate people with their boats about clean, drain, dry, keep stuff off your boat. Don't go to another lake with it until you cleaned it off. We have boot brush stations at nature centers and places with trails. The ATV in the right-hand corner there, when you come off the trail, just clean it off. Don't want to keep spreading invasives. This is what it's all about, is preventing it. So that's the quick background stuff, and we'll jump right in here to the survey and sampling. Oops, technology. Okay, real quick, you guys know this, citizen science, it's scientific work undertaken by members of the general public with or in collaboration with professional scientists and institutions there to achieve common goals. And again, can't tell you how important citizen science is. We've started calling it community science as well. There's just so much that is getting done and can get done by the general public, is getting boots on the ground, and then in organizations like lake associations and so on. So the survey we're going to propose here, the goal is to identify between 1 and 5 high priority invasive species of concern. I know you're all here most worried about Starry Stonewort, but our feeling is when you're tossing a rake out there and pulling it in, see what you got, you might as well try and look for a few others. We're not going to beat you over the head with this. If you're most comfortable just identifying Starry Stonewort, that's fantastic. If you feel like you can do a few more with some of the sources we have to learn what they are, that's even better. 1, 2, 3, 4, 5, as many as you feel like you can identify. If you see them, have them. So the basic protocol, we're trying to keep this easy. We're suggesting doing rake tosses in 1 to 3 locations, and I will have a really short, nice little video to show you in a minute all about how to do a rake toss. So 1 to 3 locations. Again, if you're most comfortable just doing 1 location, fine. 3 locations, better. Every other week, July through October now. So it's 4 months, every other week. If you do 3 tosses every other week, that's 8 weeks. 3 tosses, 24 rake tosses. So throwing the rake 24 times over 4 months, not a huge effort. We strongly suggest small groups of people, 2 as a group. It's just smart being safe around the water. That's a general thing. We all know that. We all go out kayaking by ourselves in the mornings. It's beautiful. But we're obligated to tell people, be safe, get out to a couple people. And if you're you have a lake association, 3 or 4 of you go out and you decide you'd really like to, really trying to identify more than Starry Stonewort, then you can kind of toss around ideas and discuss it and bring a guide with you and try and see if there's something there. Record what you find. Again, I'll talk about survey 1, 2, 3. I'm actually going to run through the application so you see what it looks like. We don't want anybody to be left out. If you're really not comfortable using an app on a phone or a tablet, we do have paper forms available you can fill out and send to me, or take a picture of it with a phone and send it, or whatever you want to do. But we're urging survey 1, 2, 3. This is kind of a grant requirement, but we ask you to send us 10% of the samples you take. And it's called a voucher sample. So whenever you do a survey, you look at stuff, if it's invasive, hopefully you throw it away rather back in the lake, but you're done, you move on. A voucher sample is something that you collect for further research or examination later. So we're asking for 10%, but if you do 24 rake tosses, if my math is right, 10% of 24 is 2.4 samples. So it's not real, again, we're trying to, whoops, hang on, I messed up. Okay, we're trying to keep it simple. So a couple samples, I sent out those kits to Carol. I think we included 2 or 3 little plastic Ziplocs to get you started. Uh-oh, can you still see my screen, folks? Just had a...
I can see it, David.
Yeah, yeah,
okay,
we can see you.
Something weird just happened to my end, but I guess we're good. Okay. So yeah, so send us a couple samples of just of Starry Stonewort. I mean, you can send any samples you want to me. If you get into this, some people really get into it and get really excited about identifying what's in the lake, both invasives and natives, that's fine. But we need a couple samples sent in if you do 24 rake tosses. And then I'll be checking in with you as soon as we get established who is out there deciding they can do the sampling every other week. I want to keep touch with you, and I'll try and send an email, every other week. I'd like to say I could send out some cool newsletter, but I'll be honest, I don't know if I'll be able to get that done every other week, but we'll stay in touch. And so again, we're trying to keep it simple. So you can do 1 rake toss every other week for 8 weeks, or you can do up to 3. Not difficult. We're trying to keep it simple. The point is we want you out there looking at stuff and learning how to identify invasives and native species. And it's just boots on the ground. You might find a new infestation of something, and then with any luck, we can contact somebody and you get it looked at. Okay, I am going to now, let's see. Okay, there's some pictures. I'm going to show you real quick these pictures and then run a little video that a woman that she just left our company. We're really sad to see her go. Wonderful person, but she did a neat little video about how to do rake toss. This is me. That's a solid bed of Starry Stonewort, more of it. Point these out just because you can learn about this later. You're going to have to talk about how much is on the rake, the density on the rake. So sometimes you send out and that's all you can pull in, and that's fine. You don't have to pull in a lot. It's just, and then you record the density on the rake. So with any luck here, I'm going to show this video just for a couple, it's like 4 minutes long, I think.
Today we're at beautiful Honeoye Lake in the Finger Lakes at the New York State Boat Launch, and we're going to talk about how to do a rake toss. So first you're going to need 2 rakes. You can bind them together side by side with a couple of zip ties. Connect the carabiner to the end, and you connect a rope to that carabiner. And that rope is going to be 100 feet long, and it's going to be in increments of foot between each. You're going to mark off each foot-long section of that rope. So when you do throw the rake into the water, you will be able to judge just how deep it is. When you throw the rake into the water, you want to make sure you secure the end, because you don't want it to go in the water and not be able to retrieve the rake. So you pull it out and you look at all of the plants that you have on the rake and you separate them into piles. Here I'm separating them into piles by species to see what I have present in the lake. And there's some maybe Chinese mystery snails. This is some Elodea, and that specimen there looks very bushy, but then we have another specimen of Elodea. The same plant just looks very different. So plants can look very different depending on where they are in their growth cycle. And then we have this one here, and this one is the same plant as well, only it's on the verge of dying, so it looks very different. We have some coontail, and that looks very different. That's very brown. And then you'll see there's some coontail that's very, very green and very fresh. And then some of it is much darker. So when looking at plant communities, you have to remember that one plant can look very different. It's a piece of pondweed there. Many different types of pondweed. We've got some eelgrass there up in the left-hand corner, which is a very good source of food for fish. So you always take a white bin with you and you put some water in it. And right now there's some coontail in the pail. So you take a piece off and you set it in the water. So when an aquatic plant is floating in the water, it will spread apart so you can really look at it if you need to ID it. A lot of times you'll put a coin next to it for size comparison. So if you want to send a picture to someone to help you ID that plant, they will have an idea of how big those leaflets are. So here's just another look at all of the plants. So I believe we pulled 7 plants from that one rake toss. There's 7 plants that we got out of that sample. And sometimes there's more, sometimes there's less, but you can see the different ways that plants look. So now we've got another sample in the bin, and that's Eurasian water milfoil, which is an invasive. And you see how I'm spreading it out in the water. I have the coin there for size comparison. And you can see that it has a blunt edge on the tip. It's got a little red tip. The stem is red. Those are some ID factors and when you're trying to ID aquatic plants. So thank you for watching. Anybody can make a rake for a rake toss. Take a walk down to the end of your dock and drop it in and see what you find. If you need help IDing, you could always reach out to your local prism, in this case, Finger Lakes Prism.
All right. So there's a good introduction to how to rake toss. And again, we supplied you with, I think, little white bins and enough to get some of you started on how to do this. It's not hard. It's a lot of fun, actually. It's a good excuse to get out by the water, too. Not that we all need excuses to do that. So you collected the rake, you tossed the rake out like they showed, you've looked at what you have, now you're gonna record the results. So survey 123, again, it's a free app for smartphones and tablets. We developed a form for this project. Carol, I think you're the only one that used our first one last year. This is new and improved and bigger form than our pilot we did. Why are we doing this? Well, it's quick and easy, to be honest with you. It's a great way to record stuff. There's no smudged pencil marks and wet paper and all that good stuff, and no handwriting issues. It's pretty quick and easy. It almost immediately uploads to the cloud, and then we can look at what you did and where you were and collect it, and then we can export the data to a spreadsheet for other use or whatever. We can, if any of you are interested at the end of the season, we can get the data back to you as well. We also have if it works right, we've got a few modifications to make with the live map on the Starry Stonewort website that'll show your locations where you've sampled. Just kind of a neat way to zoom in and see where you were at, and little points will pop up on the map. Won't show what you found, but it'll show where you were. And then we will, ultimately, we upload the data to the USGS Non-Indigenous Aquatic Species Database. In New York State, we do IMAP invasives, which you don't have to worry about over here. So we've been in contact with these folks, great folks. I don't want to tell tales. They're a little slow in posting stuff, but we'll send the data up. So even though I've said this is the main reason for getting you out there is to learn how to do this, identify invasive species, but the data matters. It's good. If the data is good, we're going to send it on, and it helps populate a database for everybody to use of where invasives are. So in the group of documents that you can access on the web, and I'll go over it later at the end, there's a little sheet I wrote up, page and a half, of installation and use of Survey 123. So again, this link here gets you to all the documents. I'll show it to you later. And one of those documents, number 5, is these instructions I made. In these instructions, there's this cryptic little web address, a URL. You open that from your phone, and it'll walk you through how to install the app, how to access the form for the first time, and so on. It works really well. It's easy to use. And then subsequently, you can start it like any app you would on your phone. So what I'm going to try and do now is actually walk you through the app. So this is what the app looks like. It'll look almost identical to this on a phone or a tablet. This is actually like the developer thing on my laptop here, but this way I can walk through it easily and show you. So it's 99% just like what you'd see on your phone. So you've collected you've done your rake toss, you pulled everything, you're looking at it. What do? Well, you start by, you put in your initials, your organizational name, type it out for me because I won't know it. If you have an abbreviation for your lake association, I probably wouldn't know it. You got to put where you are. This is critical so we know where you were and where you were sampling. This is the best way to do it. You click on the map. It opens up a bigger map, okay? And then you just drag, with your finger on a phone or tablet, drag over. This is where I'm at. This is actually Town Pond. And I urge you, if you notice, it had us over here to start. It actually knows, the GPS knows where I'm at. But we found out you're going to get much more accuracy if you zoom into where you are. So you know how you do pinch and zoom and roam on your phone or tablet. So zoom in quite a ways until you get to the point where you think you are. All right, and then hit the little check mark in the lower right. And that's it. And now it's logged in. You can't see it, but it's logged in coordinates, so we know where you're at. If you don't use this, you can enter coordinates manually. If you hit yes, this opens up and you add in your latitude, longitude, and decimal degrees up to 5 decimal places. To do that, you need a separate GPS unit, a GPS app on your phone, or maybe Google Maps. You can drop a coordinate and look at it. You can do this if you want. Be real careful putting the coordinates because one digit off, it can mess up things. But most cases, I'd urge you to use the map. So if you use the map, just say skip this, and it just closes all that. So do the map, coordinates manually, no, skip, good. Water body, just put a name. That's Hoops Park down the street from me. Water depth, estimate it best you can. One thing in the video she talked about is this, marking every foot on the rope on the rakes for the rake toss. We didn't do that for you, unfortunately. I think she talks about using zip ties. Don't use zip ties because you do zip tie and cut it off, and they get really sharp and they tear up your hands. So if you want to mark off the rope in 1 foot increments, use some kind of waterproof tape. Otherwise, just estimate it. There's feet and meters. And then this is where you get into what you actually found on your rake and how much you got on there. Most cases, you're going to use this rake toss on the left. Visual surveys here in case you're walking along and you go by a boat launch and there's a big glob of something that, big wad of Starry Stonewort or something that washed up. and you can say, hey, I think I should report this. So you can do a visual survey and report it. But most cases, you can do rake toss. So you click rake toss, and remember I told you you're going to talk about how much is on your rake. It's how much of everything on your rake. Don't have to worry about how much of this species is that species, just everything. 0 is no plants. Trace is a handful of plants. Sparse is 2 hands full. I'm sorry, trace is a handful of plants. Sparse is 2 hands full. Medium is an armful, and dense is covered rake. And I've had dense where you barely pull a rake out of the water. So this is for submerged, like Starry Stonewort, and this is for floating, like lily pads or something. Most cases, you're going to be doing this probably. So if you do 0, nothing really happens because the rake came up empty, and that happens, and that's okay. Because nothing means there's nothing growing there at the moment, and that happens. But if there's a trace or anything more than a trace, there's a long, nice list here of submerged species. Invasives are in red. The Starry Stonewort down here, and a bunch of others. Now, again, you know, if you want to learn how to identify a lot of these, feel free. Take your best guess at what's there. Would love to see you know, love to see you fill it out and say, oh, see, I pulled in a little eelgrass too. Let's just say. And then if you look through the water, what is submerged dominant species? Well, if you see a huge bed of Starry Stonewort, like we all have, well, there you go. That's all you got to do. Take your best guess. Do your best. If you're really completely unsure, leave it blank. It's not a problem. Okay? That's all that's there. The visual survey is the same thing. Okay? Visual survey pops this open, and you just, what species did you observe in that glob you saw or whatever. It's real simple. Okay? So do the rake toss. Floating plants, macrophyte density, okay, floating plant, you just same thing, 0, nothing happens. There's a trace. There's only a few floating species you're probably gonna ever see. Okay, you might see, you know, you might have picked up a water lily when you toss a rake. Dominant species, maybe it's duckweed, maybe there's lots of duckweed there. That's all you have to do. Sample collected, remember I mentioned the voucher sample, that's what this is for. Okay, not for the fact you threw a rake, but if you collect the sample, you hit yes. If you collect it, no. If you click no, again, nothing happens. If you click yes, it opens this up. You say, is it a voucher or other? Other is if you're really into this and you'd really like to find out what is this crazy weed I pulled in, you want to send it to us to find out, you can click that. And that's fine. You can do both if you like and send us 2 samples. Okay. The key thing is here, down here, the voucher, you want to put in your initials, right? Date. Is it still June 6, 30, 20? Sample types of voucher, right? You just put, you can abbreviate it if you want. It's number 1 or whatever. Okay. You want to make sure that you take the Sharpie and mark the bag with the same label so we know what's going on. When I see this coming in, I'm going to expect this voucher sample in the mail. It'll have this name, then I know where it came from, who it is, if in case more than one person sends one in. Other, it's the same thing. You just put in the what you think it is. Oops, other sample collected. So you just do the same thing, okay? And that's really it. You just, I think, let's see. No, okay. So I'm sorry. If you, so you do the other sample, you just put this in, just make the change. You're gonna put in that it's unknown, or you know, you're looking, what is this? You know, other, whatever, and then I'll know that you're looking for just identification, right? Then you don't have to fill this out, but notes. You say, you know, it was really windy, hard sampling my spots from the kayak, or whatever. It doesn't matter. You know, you can put any unusual things you saw in the water, whatever you want. It's just useful for us to see what you're doing. If you've got a question, you can put it here, we'll try and answer it. Then always take pictures. Use your phone. When you press this icon down here in the app, it'll activate the phone in your camera or your tablet. And take as many as you want, but take at least 1 or 2, maybe 3 photos of what you brought in on the rake. And when you take one, then you can access it plus sign, take another one, and so on and so forth. It's pretty straightforward. And when you're all done with it, press a little check mark at the bottom, and you'll see this on the phone. It's a little bit different, but it's very clear, well done. These folks did the software really well. Okay, and I'm getting an error message because I'm not on a phone, but you click that, and there you go. You're done. It'll ask you if you want to submit it now or later, depending if you have good cell signal. That's pretty much it. You know, do your best to identify what you get. That's all we're asking. Moving on. This is what it looks like on the phone. Very similar. I feel it's actually somehow easier to read on the phone. I don't know if it's because of the narrower screen, but it's easy to read. Far right here, this is that last all-important check mark to close the form and submit the data. Briefly, that's the front page of our website. But this is not a great picture. I don't know if you can see this, but this is the interactive map section. When you take a point and if all things work well, then a little dot will appear where you are sampling on the map, and you can zoom in. This lets you pan and zoom in to where you are and see. And then you see where other people are sampling as well. I'm hoping we get some people from Ducks Unlimited doing some stuff, and the folks from Indiana, we did some. These are some of the pilot areas we did last summer. Again, the voucher sample, not hard. Take a picture of it. This is a little bit redundant because you're going to do that anyway with survey 123. But if you please take a picture of it and send it to us. Use a Sharpie. Again, your name, date, location, and what you think it is. Give it a shot. What you think it is on the front. Put a question mark if you want. We'll look at it. Make sure this is the same as what you've entered in survey 123. Put a little bit of water or a wet paper towel or something in the bag. Seal it up really well, and then put it, if you're out in the field, if you have a cooler, if you know you're going to do your voucher sample that day, bring a cooler, with an ice pack in it or something. Keep it cool, get it in the refrigerator as soon as possible. Don't freeze it. Freezing will ruin the plant specimen. And then send it to us. And there's my information. Again, I'll show you what that is. When I've sent stuff before, I've just double bagged everything, made sure it's sealed up real well so the mail, doesn't get all soggy when it gets somewhere. Not difficult to do. And again, the voucher samples are most interested in Starry Stonewort, but if you have other things you want to know, if I'm on the right track, is this, some other water chestnut or something, some other invasive, let me know, send it. Okay, I can stop there for a minute before you get an identification. Questions, 1 or 2 questions now, and we'll do more at the end. We've gone through a lot. I know, Carol, if you're looking at, I know we're getting questions at the chat, but we do 1 or 2 of those or something else about
Sure.
whatever people have.
I actually had a question. The first thing I was wondering is, are we allowed to send invasive species across the border in a little Ziploc bag? That's not a problem?
You know what? You just reminded me. Maybe not. Thank you for saying that. I can't believe I just said that without remembering that. When I was talking to Nature Conservancy, somebody brought that up. And I guess it can be done with a permit or something. And when I told the Nature Conservancy was, if the permit's a quick form, sure, go for it. But if it's a long drawn out process, don't. I'll just have to tell our grantors that it's not going to be possible. So thank you for reminding me. Still doing a voucher sample specimen, if you ever send it somewhere within Canada, that's still, I guess, useful information, because that's how you do it. Generally speaking, you put it in a bag and label it carefully and send it to somebody, keeping it cold up in the last minute and ship it quickly. So thank you for that. I forgot. This is new to me. Again, international project. This is exciting, but.
Yeah, I was thinking that maybe we could send it to the invasive awareness program here. They may be able to help us. So I'll talk to them and see if that would work. That
would be great. Be wonderful. And then if you got back to us, then I could support our folks saying, this was because you can't send over the border. We've been certified by so-and-so that this is. Sure. Because we're just looking for accuracy. Send a Starry, what you think is Starry Stonewort. If it is, we're good. That's it. So.
Okay. And another question was, someone's worried that doing the rake tossing, because it spreads by fragmentation, will doing the rake tossing fragment it and cause the problem to be worse?
Boy, that's a great question. And boy, yeah, potentially yes. But at the same time, you know, for the greater good, there's this is like the established way to take surveys. And
Yeah.
Scientists and researchers do point intercept surveys where they lay out a very detailed grid. They go out there and in each point take at least 3 rake tosses, take very specific notes, and go on and on. So it is the accepted way at this point. It's a very good question. I think yes, it's potentially could spread, but we don't have a lot of other options at this point.
Yeah,
that's what I can tell you.
Something I would add to that is just make sure when you go out to do your survey, if you want to bring a bigger bucket along with you, because you don't want to throw back into the water whatever
Yes.
you've raked out of the water.
Yep.
So when Greg and I were out last week doing some of this, we had a quite large bucket and our canoe was really stinky by the time we headed back. But you know,
Yep.
just take it back with you and dispose of it well away from the shoreline. Just
Yes.
don't throw it back in the lake, and that minimizes what goes back.
Yep. And if you're tossing a rake from a dock or something, just bring it in
Yeah.
and throw it away, you know, instead
Right.
of, or, you know, a lot of places will have, we try and have safe, they're like these little wooden containers they build on land, but it's a safe place to put weeds you've pulled off your boats and stuff at boat launches. So yes, don't make the fragmentation worse than it is, if at all possible. Good point.
Someone else is wondering about where the data goes that you do collect, and is it readily downloadable? Can they retrieve the data somehow? And does it feed back to the NatureServe or Canadian Info Networks?
Again, we are sending it to the USGS Non-Indigenous Aquatic Species Database, so it's USGS NAS. They have it's kind of an old interface, but I thought it was easy to use. So yes, you can download data. You can cruise around and look at it, search a number of ways for information and data and download it. We will make it available to you folks too, as soon as we check it over if you want it. You know, Ron, we can send like a spreadsheet of what was collected. As far as Canadian sources, I don't know. USGS has told me they're very happy to connect data from Canada. I don't know if they share it with other agencies in Canada. I really don't have that information. If there's a group up there that does want it, we can work on doing that. If you know of somebody that's really excited about what we're trying to do here and get some data. I know there's, I know Brian Gincolt told me, he's asked, there's an Ontario invasive species something.
Awareness program? Yeah.
I think it's at the provincial level, I think. And he's asked them a couple times to recognize Starry Stonewort as being a problem, and he hasn't seen them do that yet. Not telling tales out of school here, but but whatever, yeah, if there's an organization up there who wants the data, we'll give it to them. Absolutely.
Okay, and someone's asking, is the Survey 123 app available on the tinyURL website?
Yes, I'll walk you through that at the end. Yes.
Okay.
It's actually within a document on the tinyURL website. I'll show that to you.
Okay, and then somebody else was saying, oops, sorry, David.
Well, I was wondering if we should just keep moving and do the rest of the end.
Sure.
Is that good? I don't want to stop you. We get more to get
Actually,
to.
maybe I'll just ask you this in case you can add it in. It's,
Yes.
It says, many of us don't have established Starry Stonewort populations, and our lakes are physically separated from each other. Could you talk about site selection for an effective early detection? And is that covered?
I would say, you know, it's good. And that brings up another point that I forgot to say. When you do your 1 to 3 rake tosses every other week, do them in the same locations. Because this is a perfect example. If you rake toss, this is great. And then you rake toss and you pull in Starry Stonewort, that means you may have a new infestation or infestation spread. As far, you know, this stuff can only spread by humans, unfortunately. You know, if your lakes are disconnected, like you said, if there's a public boat launch, that's going to probably be the first place this stuff takes hold, or any invasive for that matter. Boat launches and public access locations those are two, you know, prime places where the stuff gets established, and that's for virtually any invasive. So I would start sort of near there. You know, if you don't have too many invasives in your lake, that's fantastic. You know, and you know, doing your rake toss in 3 locations and pulling up nothing through the season is fantastic. It's good news, right? And you know, do it the following year, something comes up, well, that's bad news, and something has been introduced. So I hope that helps and not too confusing. But yeah, likely spots are where the public would put a boat in from another location. And you know, that's how it's going to get spread. Okay. So let's move on to the hardcore stuff here. Hope you're all still awake. Identification. We're going to focus mostly, as I said, on Starry Stonewort. We're going to look at these other 4: curly leaf pondweed, Eurasian milfoil, water chestnut, and hydrilla. We'll do the best we can here with lots of slides, and there's in the tiny URL spot, there's a number of references to other resources you can use to help you identify things, and online resources and so on. And I have another video to show still about identifying Starry Stonewort and comparing it to others. So we'll get to that in a minute. Okay, macrophyte. I've been throwing that word around. All macrophyte means it's a plant large enough to be seen with the naked eye, growing in or around the water, and that includes Starry Stonewort. The reason I say that is we got to remember Starry Stonewort is a macroalgae. I'll get back to that. Here's some popular references. Well, I hope ours is popular. We sent a copy of this up to you folks. Sent some copies. So we developed an invasive species guide in-house. You can access this in digital form as well. I'll have the reference for that. This presentation will be available, so you can look at it and pull up the slide if you want to really dig into these books and see if you can find them. Our resident expert in our office loves these 2 volumes. She says they're big, really well done drawings of what aquatic plants look like. Through the Looking Glass, you may have seen, it's used by a lot of folks. It's a very popular guide. And this one from the state of Maine is very good. They put it out, and it's a very good guide. So there's stuff all over the place. These are actual books you can get. And again, you know, you can, this presentation's already up on the web, and you can look at it and find the slide if you want and download this information, or we can get it to you. Basics on, I'm going to go through some basics on how to identify, what to look for to identify plants. This is just basic plant identification, aquatic plant identification 101. You need to practice. That's what it takes. Identifying plants just takes practice. So growth habit. Is it an emergent plant like a cattail? In other words, it grows up through the water and emerges to the surface. Is it floating? You know, attached by roots, or free floating like a lily pad, like you see there in the upper right corner. That's a floating. And then submersed. That's easy. Starry Stonewort, probably the bulk of the native and invasive plants you're going to find are grow on the bottom of the lakes. Identifying characteristics. These over here, leaves, and I'm going to talk about leaves in some detail. They're probably, when you're starting out, is the first thing you're going to be looking at. Arrangement, how they are, the number, how they're spaced out. Stem shape can help. Roots are important. If you get into it, rhizome, question mark here. Again, Starry Stonewort is a macroalgae. It doesn't have roots. It's got rhizoids that attach to the bottom of the lake to hold it there, but they're not roots. So they can help identify. Flowers can be tricky if it's not like a big, beautiful lily pad because they can be awful small, but it's still an identifier. Seasoned fruits, same thing. They can be a little tricky, but all these things can be used to identify aquatic plants, macroalgae. Okay, this diagram, it's a very useful, simple diagram, but it helps. Leaves, his shape, right? This is kind of the way Starry Stonewort looks. The leaves are really like little branchlets. They're not really hardly leaves at all, but technically that's what they are. You got to get the thread shape here, linear shape or something else, thicker. Wavy margins. Curly leaf pondweed has leaves that look almost exactly like this. So that's another thing. Serrated edge, hydrilla, I'll talk about that. Hydrilla, identifying, key thing for identifying is a serrated edge. Okay, that's important. Then you have all these shapes. You have these compound leaves, which can be like a single leaf, a little leaflets on it. Heart-shaped, etc., simple ovals. This gets really important over here, your leaf arrangement. Notice that, you know, this diagram part here, these are alternate. And this is trying to represent what it might look like actually on a plant. When it waves around the water, you have to look carefully where it attaches. Okay, so notice they are attached alternately. They're not attached across from each other, even though the way around the water might be hard to tell. Opposite, same thing, right opposite here, you know, it might look like they're alternate, but really look where they attach. They attach exactly across from each other. You see, I want to look at that. And whorls. Whorled, if you can see that, it's W-H-O-R-L-E-D. This is pretty key in a lot of plants. And what it is they all attach in the same spot in the stem, but they cover all around the stem. Okay, so you often talk about whorled leaves, if there's 5 whorled leaves or 8 whorled leaves or whatever it might be. Again, stems look differently from different things. We'll talk about the stems of Starry Stonewort a little bit and other macroalgae. Roots, if you're really getting into this, roots can help you identify some stuff, and particularly with, I understand that for some of the floating plants, the number of roots coming out of each one can help you identify it. Flowers, you know, flowers can be very small, nondescript things, but if you're really trying to identify, they can help you identify what's what. And seeds and fruits. I'm going to point out 2 here. This is the seed from the water chestnut, which is a floating species. These are viciously sharp. Don't ever step on one, bare feet. And they're very hard and can stay in the sediment for years and be viable. This looks like a little potato. It's a little tuber, and this is how hydrilla, which is a really nasty invasive, spreads. So in some cases, you know, these are pretty easy to identify if you find them. Otherwise, you know, some of the roots, fruits, and seeds can be a little bit challenging. So, Starry Stonewort. You guys are all pretty familiar with this. You've probably seen this diagram. Paul Skowinski from Wisconsin is an expert out there, a nice guy. He took this picture, it's been used by everybody, including us, several times. This is, you know, again, Starry Stonewort leaves are kind of, you know, look almost like stems or something, but that's what it has, and it's in whorls. Here's the point where it whorls, again, around the stem, 5 to 7 of those around the stem. The bulbils, a lot of you have seen the bulbils. These are my dirty fingernails. I'll be famous someday for this picture, but I pulled these out, these are the bulbils. They're star-shaped. If you see star-shaped bulbils, you're done. It's Starry Stonewort. Beautiful underwater picture from Scott Brown in Michigan. Aquatic macroalgae, it's not a vascular plant. It came from Europe and Asia, dumped in the St. Lawrence River, probably by boat ballast back in the 70s. Impacts, we've been over this, you know what impacts of most invasive species are. They pretty much take over, push out natives, mess up the environmental system, biodiversity, ruin, Starry Stonewort in particular, ruin spawning areas. Some species like Eurasian milfoil doesn't though, and we've always had conflicts with fishermen out here. Another thing. Water quality is a question. Brian Ginn over at Lake Simcoe is wondering, he's guessing that maybe Starry Stonewort's a phosphorus sink. In other words, they take up phosphorus in the water column based on some very preliminary data he pulled in. So nobody really knows. In Europe, believe it or not, Starry Stonewort is protected. It's rare in Europe, and they feel it cleans the water, so it's protected. So we're still wondering about that. Native species, human impacts, we've talked about that. Can't go swimming and boating through that stuff. Fishing is a problem. Potential economic impacts. What do we know? This is critical. Starry Stonewort, compared to many other invasives, we know very little about it compared to many other invasive aquatic species. As I said, there's lots of research going on right now into it. But beware that, if you have questions about it, let us know. If you get some technical question, we'll try and answer it for you, but there's still much to be learned about Starry Stonewort. This is its current extent across the region. This is from the USGS site I told you about. You can download stuff in GIS format like I did, or you can just get a basic spreadsheet you can use. So it's all over. I'm sure there's more here than shows by these dots. So you guys are up in here. So if you find some and report to USGS, ultimately they'll have this information available. It's a problem. And my understanding, it started here, like I said, like St. Lawrence, it spread this way into the upper Midwest of the US, and then came back into this area, New York State. Okay, some more slides. We're just going to drive home lots of slides and pictures because it's the best way to do this. Most of you have seen this probably, but here's some pulled right up. There's an individual long stem of it. It gets quite long, quite wide, if you will, right? Here's your whorls. There's 5 to 7 of these things around. There's little tiny bulbils, and there's some better pictures here, but down this penny, if you can see this picture, there's a bunch of star-shaped little bulbils attached to this clear, looks like fishing line. And I think Harold called it rice noodles, which is a wonderful description. They also occasionally, you can see these little orange antheridium, little growths kind of up in the branches and stuff. More pictures. You've seen all these. This is a really beautiful close-up of a bulbil. And I just learned there's a correlation that nobody understands between the number of protrusions on the star and the number of branchlets around the whorl, which is just interesting. But up in the left side, you can see these very clearly. There's a whorl with branches, whorl with branches. They're segregated out. Again, that penny picture, here it is. It gets very long. More pictures. You guys seen this stuff. This is from a harvester, a local place near here. This, I mentioned the density of a rake. This is a full dense rake. This is like almost hard to pull up onto the kayak. And this is somewhat less. Another wonderful picture from Carol, thank you. This is Carol, I've been using this picture a lot. This is a wonderful shot of a big bunch of Starry Stonewort, and then this, you can see this beautiful long clear rhizoid and moving into here and lots of bulbils all along it. It's just a really good identification picture. Okay, so now I'm going to show you Paul Skowinski again. He made this really fantastic identification video of Starry Stonewort. Look for, particularly in the beginning, he's got little frowny faces and smiley faces for invasive versus native, and he talks a lot about species that you can confuse Starry Stonewort with as he moves through this. Again, it's only about 4 minutes long, but it's a really good video.
Starry Stonewort is a species of macroalgae related to many native species in Wisconsin. This species can be challenging to identify, but learning a few important features will help you distinguish Starry Stonewort from its relatives. These algae have a simple body plan consisting of a main axis, which you could consider the stem. For simplicity's sake, we will just use the word stem in this video. Around the stem are whorls of branchlets, much like the whorls of leaves you would see on other aquatic plants. A key feature of Starry Stonewort, and probably the easiest to look for, is the presence of star-shaped bulbils. These are produced on clear threads in the sediment, which look like fishing line. A small number of native species will produce bulbils too, but these are generally round like tiny snowballs. Starry Stonewort bulbils have many points or lobes. Starry Stonewort is very large compared to most other macroalgae in our region. Notice the size of these native species compared to the nickel in each photograph. Then look at the Starry Stonewort. It is several times larger in diameter than most of our native species. Starry Stonewort produces small orange balls along its branchlets. These are the male reproductive structures called antheridia. So far, only male Starry Stonewort has been observed in North America. This means that there is no sexual reproduction occurring, and therefore no viable seeds are being produced. Without females present, Starry Stonewort can only spread by bulbils and fragments of itself. Starry Stonewort is likely to be confused with several native species. The most commonly confused species are the muskgrasses, also called chara. However, all of the common chara species have long cells running up and down the stem, creating a rough or textured feel. The stem of Starry Stonewort is smooth. Species of Nitella can also look very similar to Starry Stonewort. The branchlets of Nitella always fork at the ends, producing 2 or more equal length parts. The branchlets of Starry Stonewort may appear to fork, but what you see is actually a long bract coming out of the branchlet. Since these bracts are often only on the upper branchlets, you can look further down the stem where the branchlets don't have any forks or bracts. When Starry Stonewort is removed from the water, its stiff branchlets tend to remain in the same position. Species of Nitella tend to be very relaxed and droopy when they are out of water. Sago pondweed is a common native species that can look somewhat like Starry Stonewort. However, notice that its leaves alternate left and right on the stem rather than being in whorls. It also produces many large seeds on long stalks above the plant, which Starry Stonewort would never do. Horned pondweed has opposite or whorled leaves, but will never produce bulbils, and the leaves are very weak and delicate. It produces many clusters of 4 banana-shaped seeds along the stem, which it typically holds throughout most of the growing season. Remember, without females present, Starry Stonewort can only spread by bulbils and fragments of itself. Boat trailers, anchors, and other recreational equipment are the most likely vectors for spreading Starry Stonewort. Monitoring boat landings is a smart strategy for detecting an introduction of Starry Stonewort and slowing its spread. For help identifying Starry Stonewort and other aquatic plants of your lake, check out the field guides and other helpful resources at the UWX.
Okay. So that's, again, Paul Skowinski from Wisconsin. He's an expert. He went out 2 years in a kayak, and he wrote his own book. He has a book out there that about all upper Midwest native native and invasive species. Moving on, we're almost there, folks. Just got a bunch of slides of some of the things he just talked about and some other invasives and what they can be easily confused with. He just talked about Cara and muskgrass. A key thing of Cara and muskgrass is we pulled up, or what you think might be Starry Stonewort is because they are macroalgae, if you squeeze the stems actually pop. They do a little pop. So now you know you're at least in the ballpark. If it has a garlicky or skunk-like odor, it's muskgrass. Okay, so then you know that, okay, this isn't Starry Stonewort. It's not overpowering, but it's definitely there. It's an interesting smell. That's another way that I've done a lot. You pop the stem and smell it. If it's stinky, then it's muskgrass. Sago pondweed he mentioned. It's kind of, I find this kind of feathery, right? It's one stem might confuse you, but it's pretty easy to tell apart from Starry Stonewort because it is the kind of the brush-like feathery. You know, here it says it's kind of fan-like. So it's quite a bit different. And then there's those little seeds on the ends that he talked about. So that's Starry Stonewort and some lookalikes. I just want to do some other invasives. Curly leaf pondweed. This stuff grows off the bottom and forms these big mats on the surface, as you can see here. The leaves, remember the leaf diagram, this is a pretty easy one to identify because it's got these oblong kind of stiff leaves. They kind of got these wavy margins, they call them. I guess it's the technical term. It's wavy and lobed on the end. They're kind of blunt. They're not sharp on the end. They're rounded, kind of blunt. You see it in this picture up here. Pretty easy to tell. I guess they do grow little flowers on occasion. But this is, they grow up and they float across the surface. I don't know if they grow across the surface. I'd have to ask somebody, but they grow up and they form a mat, but they're attached to the bottom for sure. Eurasian milfoil, the bane of all of our existence. This stuff is everywhere. Key thing to remember is it's almost always has this red or brown stem. Okay, and sometimes the edges of the still there? Okay, the edges are can be tinted in red as well. Okay, and they're also kind of blunt. They look kind of like somebody cut them with scissors, so they're almost squared off here. Okay, that's another thing to see. Each one of these things, there's 4 of each one of these in a whorl. Remember I talked about the whorls? There's 4 of those around a location, and then each one of these has at least 12 pairs of alternating leaflets. Okay, so it gets pretty complicated, but so 4 of those, and then each one of those made up at least 12 opposite things. So often confused with common coontail. Coontail is a very common, very common native plant, great plant. It looks like a raccoon's tail. You notice it's very similar, but it's all green. There's no red, and it's a little pointier. You know, if I can go back, causing problems. Go back, these again, these are kind of squared off, you know, like squared, this is kind of square over here on the top. Whereas coontail's a little pointier in each place, pointier on the end, and it's all green. Hydrilla. Hydrilla is it's suspect number 1 down here in New York State because it's not here that much yet, and once it gets a hold, it's very aggressive. So that hole, if you remember way back at the beginning, I looked at that graph. Now is the time they're finding little tiny new beds of the stuff. They're able to eradicate it with chemicals or hand pulling, whatever it is. It looks very much like Elodea. There's several different species of Elodea, both native and invasive. So I'm not going to get into identifying Elodeas, but I do want to talk about telling Hydrilla apart from Elodeas. And the key thing, I know it's small, but right here on the edges, there's that serrated edge, okay? There's usually 5, but it can be 3 to 8 whorls around here, but the serrated edge tells us apart from Elodea. Okay, and also if you do pull up out of the sediment, there's these little potato-like tubers, and that's how it spreads. So if you pull this up, it could be Elodea, but look very closely with a hand lens or something, and you'll be able to see if it's serrated as Hydrilla, and then you want to definitely take action if you can and take care of it and get it out of there. So here's a species of Elodea. Again, see, it looks, if you can see, it's hard jumping back and forth, but see, you get all these. This is only got whorls of 3. They're kind of the same little leaves. They kind of grow off the stalk. I'm gonna go back. Whoops, I went forward. Sorry, folks. Okay, same kind of, looks very similar, but it doesn't have the serrated. And this has, Hydrilla has more in the whorl than the species of Elodea. So a little hard to tell apart, but it's there. Water chestnut, this is a floating, this is really easy to identify. This is probably easiest. It's a floating invasive. It grows in these I forgot what they call them. These, I apologize. But these groupings here of plants. Does it show me what they call them here? It doesn't say, but they're in these groups. So there's like a common, you know, a cluster of roots that goes down from this. You can pull this out. There's somebody holding one in each hand. Okay, they're quite large. They float. They have these strongly serrated rectangular or triangular leaves. And if they get infestations, we've had them here in New York State, they completely take over a cove or a small end of a pond or lake. You can't miss them. These are the seeds down there. You don't want to step on those. Those are the seeds. So if you see this, the good news about water chestnut is we have had luck eradicating this from areas. It takes several seasons. You get a whole bunch of volunteers in canoes, and you hand pull it. And it's easy to pull, or if it's shallow enough waders, you pull out tons of this stuff. And then each season is less and less, and over 3 or 4 seasons, we've been able to eradicate it in small areas. Really easy to identify. Eelgrass, this is just a couple good guys now. Eelgrass is easy to identify. It's a good fish habitat and food, is my understanding, and it looks like grass. It looks like just tall grass growing in the water. Really easy to identify. Duckweed's fun because it looks like little tiny lily pads. At first glance, some people think it looks like an algae bloom, but it's not. It's duckweed. This is common duckweed. There's a different duckweed called greater duckweed that I guess has got a purplish red underside and a purple red spot on it. Again, if you get really into identifying this stuff, look for that. But duckweed is an interesting plant and a native. So that's it, folks. We're going to talk real quick again about the resources. This is the address to find, and if you haven't been sent to you, we can email it to you. There's a certain document I'm going to show you in a minute with links to further references. There's all this information there. There's a whole bunch of stuff, and I'm just going to show it to you rather than talk about it. I'm going to sort it. Hang on. Okay. So this is what's there. There's a lot of references. Again, this is all that tiny URL site. Okay, we've got a folder with fact sheets. Curly leaf pondweed, milfoil, hydrilla, starry stonewort, water chestnut. We got I've pulled these fact sheets from all sorts of resources from different states and things. They're good to look at. I find that looking at fact sheets from different locations and organizations and states, it's all the same plants, but they might take a different photo or describe it a little differently, and it helps to read multiple ones. We got a bunch of the Starry Stonewort that we've collected, and at least a couple in each of these. Okay, so I won't go through all these now, but you can look through these. It's a mission statement of why we're doing this. A couple of documents about the Starry Stonewort Collaborative. You want to pass those on to anybody that might be interested. There's kind of an overview of what I've just told you about now for over an hour and a half. It's just kind of a general discussion about the survey program. This is the one, number 5. I'm going to open this document. Somebody asked this question. If it'll open. Oh, come on. Maybe not, folks. Hang on. There it is. Hope you can see it. Okay, so this is a little 1 or 2 page document I wrote up that kind of walks you through how to install and use Survey 123 application. Here in the middle, it says access the URL, the web address. This is how you get to it. You access this web address from your phone or your tablet, and it'll walk you through how to install and how to use the form and everything. It's all right there. And of course, I'm available for questions. Most people who've used this haven't had too much trouble. We haven't had a lot of problems. It walks through it pretty cleanly, and we tried to write up these instructions to be fairly straightforward as well. There's a plant surveillance and ID guide from our own New York State, which is pretty good. There's another one we were able to get a draft of from a private consultant. Just more information about how to identify and pictures and things of different plants and invasives. We threw something in about harmful algal blooms are a problem down here, so I don't know if they're up there, but everybody should know about them. There's the data sheet. If you feel you really can't use Survey 123, there's your paper data sheet. And then this down the bottom, this is this actual PowerPoint presentation you just saw, okay, or a general version of it. And then this one, number 10, good old 10. Okay, if you pass this on to somebody, they can get right back. There's the teeny URL again, tiny URL, there it is. Another place to access the presentation, okay? Now, the videos I showed, okay, you can get to them through the Starry Stonewort website, or you can try and get them directly through YouTube. I find it easier to go through the Starry Stonewort website at this location because they're right there. And then there's a whole bunch of fact sheets and more information at this website on our own Finger Lakes Institute PRISM website. And then the field guide we sent copies of up to you, hard copies. It's at this website, and some more information as well. You can go in and print just a few pages if you wanted to, or just look at it online. And then one more resource, we set up this Gmail account. If you see something you're just interested in outside of the program and just want to send a photo and say, what is this? We'll help you ID it by sending that photo to that location. My contact information is on here too. My email, phone number, leave a message. With COVID-19, we're all working from home, but if you leave a message, that's forwarded to me. And then here's the mailing address at the office in Geneva, and I will get that eventually. If you do send in the voucher sample, please email me so I can make sure it doesn't sit there stinking in our office. I'll run in and get it, the office, and pick that up. So again, this is document number 10 with further links to resources and contact information. And of course, you have all these. Okay. And with that, we can take questions. I'm going to ask you again, work for a minute. I'm going to send out a poll of 4 questions. Make sure you give an answer for all 4 of them. You'll see what I mean. You get number 3, but make sure you do there's none of the above and 3, but do answer all 4 questions, or else it won't submit properly with Zoom. I'm not sure why. So in a minute or a second, you should see a poll come up in front of you, and just click on the answer you want for the 4 questions. I really appreciate this. And then we'll take questions. Is everybody seeing the poll, or is anybody not seeing the poll?
Yeah, Greg sees it, but I don't.
Okay. Did you say you're on a tablet?
Yeah.
I'm wondering if it's coming up, like if you swiped to another screen, you might see it. I'm not sure.
Poll in progress.
I'm not sure I can help you with that.
Oh, here it is. Yep. Okay, thank you.
Lucky guess.
You're just so smart. Okay, it says host and host and co-host can't vote. So
Oh, you can't vote? Okay, well, make sure.
that's okay. I'll give you a
Greg knows
thumbs up.
what you like. Make sure Greg votes what you want to vote.
Yeah. I was gonna say he does that anyway, but I don't.
No
really
pressure.
wanna say that. Get myself in trouble.
I don't know, Carol, I don't see anything. So I see questions, feedback, poll, David's email address, and 2 ducks. So what am I doing wrong?
I'm not sure.
I guess I'm
Not sure, Patty.
You kind of broke up. What do you see? I don't know if I can help you here.
I think she's just seeing the slide that you have up with the ducks.
the slide, right, that's all I see.
Are you on your iPad, Patty?
Yes, yeah,
So swipe down from the top. It says poll in progress.
yes, I see that.
Okay, now click on the poll in progress.
Okay, just poll in progress, yes. Oh, okay. There it
You
is,
see-
thank you.
Outstanding.
Yeah, I
Thank you.
got it, it's fine. Yeah.
This is good. See, I'm learning too, so I can give better support to folks on tablets.
Well, yes, I'm very happy to help you in this regards.
Well, I appreciate it.
Oh, cool.
Okay. See, I can tell. I get 10 of you participating in the poll. You all don't have to do it. If you can, I'd appreciate it. Let's see, 2 of those are Carol and I, so it's.
So David,
Yes.
you've talked a lot about identifying, and I know this is a tough area. What's the latest on trying to eradicate Starry Stonewort down in your area?
There's eradicating, nobody knows how to eradicate it yet. But probably the most popular thing happening down here is harvesting. And harvesting is only a Band-Aid, although there's a local lake association to us that is really interested in measuring what they harvest, because anecdotally, in other words, not scientifically gathered information, the harvester driver is doing this every season, and he seems to think the beds are getting smaller and smaller every year. So whether that's true or not, we don't know, but they're looking at what are the best ways to measure biomass or weight of what you've harvested. If you do that every year, maybe we can tell something. So that's one method of control, not eradication. New York State is legendary for how hard it is to get a permit for chemical application. I'm not saying it's a bad thing, but when I met with people from the Midwest, they go, "Oh yeah, I've heard of New York State," because they were contractors that would love to get a job out here. So in the Midwest, there's been, in Michigan in particular, a tremendous amount of work applying algicides and stuff to eradicate it. And it's not, it knocks it down, I believe, for a while, but they can't completely kill it. So there's not a lot of good news there, except there's a lot of as I've said a couple of times, an awful lot of research going on. Michelle Nault from Department of Natural Resources in Wisconsin, if you caught her presentation, we did a webinar with a presentation that you can also find on our website. On their table is one method of controls to do nothing. which is frightening, but they've seen one lake in particular. They've done nothing for years. It's a small lake, and they're seeing the Starry Stonewort beds actually reducing in size. But nobody knows why. There's no scientific reasoning for that yet. So everything's on the table. Eradication right now, nobody's figured it out, but there's just lots of control methods to figure out, which help. If you harvest it and get a boat through there now, that's great. But it also hopefully will feed into learning more about how to eradicate it someday. If that makes sense.
Hi, David. Oops, sorry.
Yes.
When you're talking about the harvesting, the one thing you have to remember is that this is spread through fragmentation. So you want to be very, very careful how that's done.
That's true. Absolutely true. You're absolutely right. And it's, they've been doing it at this one lake association, and it's one of the other Finger Lakes for a long time. And they are concerned about it. But at the same time, it would virtually, if they did the harvest, virtually shut down a boat ramp in this one area. It's like the one boat ramp in town, and one of the primary on the lake and only one in their town. So yeah, it's a problem. How do you balance it, economic loss versus spreading it further? Fortunately, it's kind of like an inlet. It's like an inlet channel, so it's hopefully being contained more or less there and not flowing too much in the lake. But you're right, it's a problem. You gotta be real careful with it.
There's a couple of questions here. Someone has downloaded the Survey 123 app by clicking on the tiny link, but it is asking to sign in to an ArcGIS online username password required. Do we have a sign-in account?
No, boy, that's not good news. You shouldn't have to have one. Did you download it from whoever did that directly from your phone?
I'm thinking probably.
Or tablet?
Yes, this is Brett here.
Hi, Brett.
I followed the link, and then it asks you whether or not to download it on Google Play Store, and you click that, and then it downloads, and everything is good. And then when you open it up, it says you don't have any surveys installed. Would you like to install one?
Yep.
Type of thing, and then it wants you to connect to an ArcGIS server with a username and password.
Oh.
But
Okay.
you can sign in with Facebook or Google. You can sign in with your Facebook.
You shouldn't have to sign in at all. That's the point of this thing. So my very great apologies here. I don't know what's going on there. That'll be project number 1 this afternoon. I'll find out that's what the URL is for, is to bypass all that. Because those of us, the college where I work, we have licensing to build these forms and all that kind of stuff. That's what that's for. And when you push it out publicly, you're not supposed to have any login at all. So I apologize. I'll fix that as soon as I can. I don't know why it's doing that. It's not supposed to, but I will find out. So apologies, and I will email everybody when I got that rectified. Thank you, Brett, for finding that out, though. Okay.
Okay.
I'm going to stop the poll. I think we're all done with the poll.
Oh, Anne had said she couldn't submit the poll.
No.
So is there something?
It's okay, I got it.
Okay. All right. Thanks, Anne.
Thank you. Okay, I'm going to end the poll for whatever that's worth.
Yeah.
Yeah, I'm still learning how to use Zoom. I don't trust, I like Zoom. It works really well most of the time. There's too many options. Okay. Okay, any other questions? I can focus on questions now. Just put up, I urge you to go to the website when you get a chance. There's lots of information about the program, links, the Facebook, if you're into Facebook.
Huh? I don't see the white flower. No, see, I've never seen the white flowers. We never talked.
Okay. Any other questions? You guys are easy.
Greg, got any questions?
I have a question.
This is Diane Trousey. Can I just ask a question without typing it in?
Sure, sure.
So I was reading some Starry Stonewort publications, and it was saying that Starry Stonewort is, going like crazy in North America, but in its in its native range, it's actually started to become a species that is endangered. And they're actually talking about trying to introduce it to improve water quality.
Yes.
Any comments on that?
When I first heard that, I was floored. I mean, when I first got into this, it's, yes, you're absolutely right. It's crazy. One
I know,
thing,
I was floored too.
yeah, yeah, I know it's protected over there. I know I'm not sure about Asia, but I know in Europe, Northern Europe and Germany and things, they're really trying to protect it, and they feel it cleans the water. So the big question is the why, right? Why is it not growing there and grows out of control here? And the experts are looking at that. And one thing that may be is our waters here in the northeastern U.S. and where you guys are in southern Canada, you know, our waters are calcium rich. And one thing that is in agreement with some of the experts is that calcium down, you know, commonly you might hear it called hard water, you know, calcium rich water. It's a limiting factor with Starry Stonewort. So one area is to maybe look at some of the water in Europe and Asia and see if they're not calcium rich. Maybe that's the limiting factor. You know, maybe that's the thing. You know, what we do about it, I have no idea. I wouldn't know how you buffer out calcium in a lake. You know, because that's, you know, we have bedrock and other things here. We're just calcium rich in this region. So that's maybe one thing. It's possible, you know, there's areas of Europe, water quality in Europe is, you know, I haven't traveled to Europe myself, but I've heard that sometimes the water quality there is horrible compared, you know, we're blessed with good water here in the Great Lakes region. And, you know, maybe the water's dirty enough out there that it can't grow. I don't know. Maybe they'll put it in there, introduce it, to clean the water and then take off. I really don't know. So it is a big mystery. I know there's some experts looking at that. I have just heard that there's some agreement around calcium carbonate, I guess it is, in the water column. That maybe what's going on.
Thank you.
Welcome.
You'll be fine. It needs air. There's no air in the water.
Anybody else?
Hi, Dave. I have a quick question. It's Brett here.
Yes, Brett.
Just in terms of the sampling frequency or the intensity, I know you're asking for a commitment of, you know, the same site every other week. Yep. And then just to clarify, you had the 1 to 3 samples.
Right.
And is that 1 to 3 tosses of the rake?
That's 1 to 3 locations. So like if you, I don't know, if you've got waterfront and you go out and you've got 3 nice little inlets you really like to go in your kayak, take those locations, go to each site, toss the rake, record it, survey 1, 2, 3, go to the next site, record it, survey 1, 2, 3. Does that make sense? So one rake toss per site.
Okay.
Does that make sense?
Yeah, no, thanks for that. And then if for example, I couldn't sample every other week, right? Like, just say I wanted to do kind of more like a reconnaissance survey around a lake. So multiple sites, but only once, not, for example, every other week through July and October. Is that data that, would you discourage the use of this app for that type of sampling? Or would you encourage that, even though it doesn't fully meet the intent of the protocol, because we're just getting more data on Starry Stonewort?
No, I think you could use it. I mean, if you remember, when I went through it quickly, there's a visual survey. Is that what you meant? You go around the boat and do visual surveys? Or would you be tossing rakes?
Cheese and crackers and the soup. It's out in the freezer. I got a thought.
Patty, you're not muted.
Brett, when you do that survey around the lake, would you be doing rake tosses or just visual surveys in each location?
Well, it's to be determined, I guess. It all depends on if the infrastructure is kind of set up to, record the data.
Mm-hmm.
But
Okay.
just from looking at expansion opportunities down the road, and once this infrastructure set up, it might be quite easily to get a lot of people to do this, once or twice, rather than, sign up for the full commitment of every other week throughout the summer.
Yeah, no, I mean, I don't see why you couldn't use Survey 123 to do that. Just whenever you stop, and if it's visual, type visual. If you toss a rake and put that in into the system. If you're going to do that, maybe just send me an email and tell me your general plan, so I know that data's coming and it's going to be a little different from everybody else's. You know what I mean? But I don't see it being a problem. It's we're not trying to be overlords here. We just want to get people out there doing stuff and in some kind of process. But if, when I talked to the Nature Conservancy and Ducks Unlimited last week, they were talking about there's one, I can't remember who it was, but there's a couple lakes that are so remote, these locations, they can only get to like once a season or something. I said, sure, do that, just let me know. So no, that's fine. I would go ahead and do that. Just maybe drop me a quick email saying this is what we're going to do. It'd be helpful.
Thank you.
Okay, folks, we've got a long time here. I really appreciate y'all sitting in. If there's any more questions, I'll take them, but I don't want to tie you guys out too much longer.
So hi David. So you're absolutely fine that if people on some of our lakes that hopefully don't yet have Starry Stonewort do the program and they don't find anything, you're good with that, right?
Sure, absolutely. I mean, it's just, if they pull up some other invasive, let us know. Again, that's why we're urging you to look at some of the, some of the documentation stuff and maybe identify some other, if there's Eurasian milfoil, to let us know, for sure. But yes, if there's a negative result, it's still important information.
Right.
If it's at one end of a lake and not at the other end, it could be valuable to survey at the other end where it's not yet, and hopefully it won't get there. But if it does, it shows it's spreading quickly or spreading quickly. Yep.
So David,
Yes.
if we can send the plastic vouchers across the border,
Yes.
we do it with sending you a picture.
I would say that's probably our only option, but yes, let's go with, I mean, if you use Survey 123, probably take a picture anyway, but if there's one in particular, you say, this is my voucher, and take if you remember that video from Patty that she did the nice, how to throw a rake toss, put a real nice picture with a coin next to it or something as best you can, and send that, say this is a voucher picture. And I will find out, or not find out, I will tell our folks here that that's all we can really do, because there's but I think that would be fine if it's a nice clear picture, and maybe you could fill out a piece of paper with the information next to it or something, and just frame up a little picture that shows as long as we can identify what it's in. If the picture is nice and clear, we can usually identify what it is. Does that make sense?
And just a reminder, I'm going to talk to Brookshire at the Ontario Invasive Species Awareness Program and see if he'd be willing to, at least collect the vouchers and say, yes, that is Starry Stonewort.
Well, that'd be fantastic if you could.
Yeah, I'll ask. And,
Yes,
and if he doesn't want to do that, I would be willing to probably collect them as well, so I can see that it actually is Starry Stonewort.
Okay, that'd be
But
great.
we'll work out something.
That's all we're doing. It's just a quick check. We're not talking anything major. Just here's my Starry Stonewort sample. If it's not, then we'll email you and say, okay, that's not quite right, and talk about it. But so yeah, absolutely. And go ahead, Carol, CC me when you talk to Brooke.
Yes, I will. Yeah, he wanted to reconnect with you anyway. So
yes, fantastic. More connections.
Yep.
David, thank you for your presentation and your time. One quick question. How is the work going on the best management practices that you mentioned earlier?
It's still at the research, and it's still at the research level of what we call control, how to do it. As I've said, there's people, in the Midwest that are done. Somebody was trying diver assisted suction harvesting, which is, if you don't know what that is, you get a diver in this big, basically a vacuum, and they suck up all the plants underwater. And that's they're trying to see if that's useful for small infestations. Harvesting, again, they're looking at to see if long-term, over a number of seasons, that's having an effect. A lot of chemical application research being done. I know that, again, Brian Ginn over at Lake Simcoe, you know, he talks about in his presentations, you know, they eradicated Eurasian milfoil with dicot, I think. And as soon as it was gone, it just filled in with Starry Stonewort over just a couple seasons. So, you know, that's another thing, Starry Stonewort knows it really likes disturbed areas. So there's all this information swirling around. There is on July 22nd, Dan Larkin and his associate are going to do a presentation. We're hosting a webinar, sorry. And that information is on the event calendar on our website, or we can get it to you. He's going to talk about the effectiveness of control measures. And he's looking at data from 3 different states. So it's a pretty neat, they're doing a pretty big analysis of what's effective and what isn't. So again, best management practices, there really aren't any, and it was something we really wanted to do for the collaborative, but so what we're doing now is just helping promote and support any kind of research on controlling the stuff. Because you get to the point where you control it where it goes away, then you've got a best management practice. So that's all I can tell you. It's not good news, but we're working to get there.
Okay, so I guess the next thing is, now that everybody's seen the training webinar and have an idea of what's involved in the program, if you could let me or Bill Napier know if you're interested in pursuing actually doing the rake tosses, just, you know, maybe one little group on each lake to start with this year, 1 or 3 sites, that would be great. And David, as he mentioned, has sent me his survey kits, which I have in my laundry room, just waiting for anxious people. So if you let me know that you are interested, I will get you that survey kit, hopefully either this week or next. I'm more than happy to come around and deliver them to you. So that's my plan so far. So if you could send either Bill or I, or Bill and I, a quick email to let us know that you're interested, that would be great.
Or they can come and visit us and see what an infestation looks like.
Or my husband just suggested, or you could come to Stony Lake and come and see our lovely giant infestation.
Show me pictures. It looks, I mean, not Starry Storms now, but it looks beautiful. I would love to come up and see that. But
you can come anytime.
I need to, I got to make sure my license is the right one so I can get back home. I think I can get into Canada, no problem, but I don't know if they'll let me back. I gotta make sure I have the right license. I think I do, because I don't have a passport yet, or at this point. So.
Oh, well, you'll need a passport and an open border. But as soon as you have those 2 things, you're more than welcome anytime.
Well,
you can come and wallow in our Starry Stonewort.
Yeah. There's one more. I just popped open. I haven't been looking at the group chat. I just popped it open. Carol, what's from William Napier to everyone?
Yep.
Did we answer that?
That was sort of what I was just mentioning, that we'll need people who want to do the survey to contact Bill or I, because Kawartha Lakes is going to take on keeping us organized, so that would be great.
Wonderful. Well, this is great, everybody. I really appreciate you taking time. I know it's a lot of information quickly, but you know, look at it again. You know, again, we got a lot of the resources. You can look at this presentation again or one like it. The videos are there. We're here for questions, so hopefully we get more boots on the ground looking at this stuff. That's what it's all about, is early detection. You know, if you folks talking about, you know, throwing a rake and not getting Starry Stonewort, that's great. But if there's, you know, a couple spindly pieces of hydrilla, that's a real problem too. So it's all about early detection, getting more people out there looking for stuff. So, all right.
Yep.
If no more questions, I guess we're good.
Okay.
Thank you all. Whoop. Do we have another question?
Nope, that was just a thanks.
Oh, that's nice.
Thanks.
Thank you.
Thank you, Carol and David.
Okay, thank you very much.
Enjoy the day.
Yeah.
Hope everybody's surviving the pandemic okay.
Oh, yeah. wearing our masks.
Yeah, seriously. All right. Carol, you want to chat here for a minute?
Sure.
People drop off here.
Yep.
Yeah, it's getting humid here. I don't know what weather you've got, but we've got a beautiful stretch of weather, but it's getting steamy today.
Yeah, I live for the steamy weather. That's why
Oh, do you really?
I spend so long cold.
Yeah, I know. Live up here in this region. It's chilly. But, so you and I will be in touch.
Yeah.
Carol, I mean, you became the unofficial collaborator here.
Sure. I'm just wondering who Pixel is. I see a participant that says Pixel. Who is that?
I don't know. Could be a phone. And they might, because it might be a phone name. You know.
I'm still following, Carol.
Okay.
Doug Colmer.
Who is that?
Oh, Doug.
Oh, Doug. Oh, that's Doug. Okay. Sorry, Doug.
So anyway,
my phone signed me on. My phone signed me on as Pixel, and I'm kind of just waiting for some of the numbers to reduce to actually have a chat with David for a minute.
Oh, okay. Well, hang on then. So Carol, we'll talk. I have emails for
You
everybody.
don't want to do the same thing, Doug, so.
Well, there's only, I see 6 of us on. If we want to have a discussion now, that's fine.
Sure. Yeah, go ahead.
Well, before Doug jumped in, I just had a couple of observations around this whole harvesting issue. It's Patty McDonald, Carol's neighbor.
Yep.
Yeah. And up here, especially, and Brian didn't mention it specifically in his presentation, but I talked to the people in Romaro where Carol and Greg happened to be paddling last summer, and I got a hold of the counselor to sort of get some history as to what had happened there. He said that at this point, they're currently taking out 16 tons of that stuff a day. And so when you get to the point where the harvesting is at that level, I don't know, Doug, about the lakes up there, but around here, none of our cottager roads, would even support that kind of operation if we could even get the townships and others willing to do it. So the whole harvesting issue, it seems like where people around here are harvesting, and I think Scugog had a similar issue, it just gets so massive, it's just not supportable. So I'm still trying to figure out this hand harvesting thing and what's too big to actually do and what might make it worse rather than better. But Carol and I have had several conversations around this, and we've got lots of spots very close to both our cottages where it's really quite shallow. And as we're looking at the growing, I think, a bit more slowly than I think it was. And it would be possible to get in there with waders and canoes and actually try and get out some of this stuff. And since it's already inundated the entire area, maybe I should be, but I can't decide whether it's worth the effort or it would just be a frustrating waste of time and it would just make it worse. But we do have several areas where either right now we can see it's very, hasn't really started taking off yet. So if we got some divers, which we do have locally, we could probably get some people down there. The dash suctioning is probably not in the cards this year because of what's involved. But your feeling about trying to play around with a few ideas, because to me, we've got a living laboratory of this stuff that nobody seems to be all that interested in looking at. So I decided to ask you about that before I signed off. And it sort of tied in what Doug was asking about, best management practices and that sort of thing. So.
Boy, again, hand harvesting is being tried. And diver-assisted suction harvesting is kind of the same thing on a grand scale because using a giant vacuum and divers, but it's still being tried. There's 2 key things, like, as I'm sure you want to try and reduce the amount of little segments that break off and float away while you're hand harvesting, right? You know that.
Right.
You want to control that. And the other key thing is, my understanding from the experts, you want to make real sure when you're doing the hand harvesting, you try your best to dig down into the sediment and get those little bulbils. Because if you just tear off the plant and the bulbils stay in the sediment, it's just going to grow right back again really quickly. So one of the keys to hand harvesting is getting down in the sediment and pulling up those little bulbils as best you can as well. I'm going to guess, and I'm not the expert here on this stuff, but I'm going to guess that if you hand harvested a small area repeatedly, you know, every summer or whatever it is, and it's going to slow down that area. But again, you're going to have to make sure you keep the fragments to contained and really get down and get those bulbils out of the sediment if you can. But it's an unknown still. You know, that's my guess. You know, they're still trying to figure out. So is it worth the time? You know, there's a time sink. You want to be careful. You know, of course, we don't want to promote people getting in the water, getting in an emergency because the waders accidentally fill up and they fall. You know, whatever it is, we want to be careful.
Yeah, yeah. No, yeah. What I'm talking about, it's very shallow, and it just seems like it.
So I would, yeah, you know, you're not, as long as you can keep the fragmentation down, you're not going to hurt anything, right? You might make a difference. You know, if you can document it even loosely, you know, that you did it 2-3 summers in a row and there seems to be less now, that's positive information. It's just very, as you know, or you can imagine, it's very time consuming. And suction harvesting, no, okay. Suction harvesting probably is more effective, but you know, now there's an expense in divers and equipment and all that stuff. So I wish I could tell you more. I wish I could give you the silver bullet and say, yeah, do it. I mean, you're not gonna hurt anything.
Yeah.
But you may
Well,
be right
again, I
around your
just
goal
don't
launch,
want to make it worse. And I've been thinking about it now for the 3 years, and we haven't really done it. There's been different reasons why we haven't, and we watched the last webinar from Wisconsin and saw all of the different things that have been tried, some of which, you know, we haven't done anything with up here. So that was all helpful. But yeah, the other thing I'm noticing is right now, there's a lot of decaying weeds where the Starry Stonewort is.
Yeah.
No.
Underneath it is the very healthy Starry Stonewort, which didn't happen last year or the summer before. This is something that's a bit different. We've always had weeds decay in my bay, but usually it happens in late August. It's the end of the season, you know, sort of, and weeds have, you know, they've sort of run their course, and so you get these big balls of decaying stuff that I pull out. But this is much earlier. This isn't even July, and it just seems to be maybe that the word is just crowding out what weeds are there to the point where they're decaying. I don't know.
Yeah. You know, I don't have an answer for you. I know in some places, man, I don't know, maybe Carol, maybe you mentioned this, I can't remember, but there's places that Starry Stonewort will grow up, it'll break the surface in the fall and then have this huge die-off all at once, which is unusual. I don't think we've ever seen it down here, but it happens up there, and I know it happens some places in Michigan, so it's another unique thing. So what you're seeing now, I don't know if last year's Starry Stonewort or something else, I really couldn't tell you. I do know down here anyway, Starry Stonewort started growing later, and a fellow from Michigan I know I talked to says it's growing, it's not as big this year. You know, we still had snow flurries in May here. It was pretty awful spring. So water stayed cold, so the Starry's growing later here. I don't know if that's connected somehow. I guess I couldn't tell you what the dead vegetation is from. Could be just left over from last year.
I think we should move on and maybe let Doug ask his questions.
Yeah.
Thank you. I had a question to start with Patty and Carol as to what luck Wildfire Golf Course had with their experimental project last year. Have you heard any feedback from them? They spent thousands in making an effort to try and get rid of the weeds in front of their place with where their docking facilities are for people coming in to play golf.
I would like to know what their
You've been in-
experiment was, because that's something we've been wondering about. Chemicals are not allowed, and I checked with Trent Severn, there's no permit.
No,
So
they did
if
not
you
use.
know more,
They did not use
I'd like to
chemicals.
talk to you offline.
They did not.
about that.
I can tell you what they used. They used
Yeah,
Weed
can we talk
Begone weed
about that
mat.
off? They used what?
Pardon?
What did you say they used?
They used a company in Coburg called Weed Begone, who comes into marinas and boat launches and places and puts down a matting situation to control out of control weeds.
Okay.
And according to them, they've been very successful.
Did you say they
Yeah.
use
We'll
mats?
we'll
Stone-
talk about that after, Doug. I'll talk to you about that one after, if that's okay with you.
I'm asking David if he has any experience with weed matting being tried, or whether they even have access to a company that supplies weed mats.
I don't, I know some places are trying weed matting. It is one of the many things that people are trying. This one, boy, I think he was Nature Conservancy in Indiana, and this is like a year ago. I got an email from him. We conversed via email, and they tried some weed matting, and it seemed to work, kind of. But the jury was out still, because sediment and things fell down on top of the mat, and then Starry Stonewort started colonizing on the sediment and stuff on top of the mat, which is kind of crazy. But so it did work kind of in some areas, but he felt, so it was maybe a viable solution. But again, the jury's out how well that works. And of course, you're going to suppress anything too. That's the other thing, is that as we suppress the invasives, we want to try and keep the natives going as well. And, so I don't have a clean answer for you. It has been tried. It is being tried with some positive results. But I'm not really familiar with any companies that do that.
Okay, I'll just mention that there was an effort made in Quebec by the environmental people there, and they hired companies to go in to their provincial parks to try and control milfoil, and they were using the propulsion systems, and they were using matting, and they were using some type of algicide, I believe, as well. And it was actually being sponsored by the Quebec government, and there's quite a lot on the internet about it as to what that particular effort was done, but that was more in an effort to fight milfoil than starry.
Right. And don't forget, I
But
got to
I'm
too, just real quick, with any of these things, one thing that has been documented is Starry Stonewort really likes disturbed areas. So if you do hand pulling or whatever your method is, or even matting, you got to stick with it, because there's a good chance that you knock down whatever's there, and now it's got from another location. The Starry Stonewort is going to go, oh goody, I got some disturbed area, and come right in and repopulate.
Well, the idea is, if you are going to use matting, you put it down without disturbing the area. Because if you just leave it grow and then try to handle it with harvesters later, then that's like setting a bomb off in the bay.
Agreed. Yep, you're right. So matting, it's got some positive results, but as far as I haven't found or seen a really definitive report on it, how well it controls Starry Stonewort. It's not that it can't, I just haven't seen it that much. And again, this one guy did it, and there was some evidence that the starry was starting to recolonize on top of the mat as soon as some sediment fell on it. So I'm not sure how you control that. But it's interesting.
It is much easier to control the small amount of starry that would accumulate on top of the mat than it would be to try and control it with a harvester.
Yep, absolutely. I find it very interesting.
Okay, the comparison there is night and day.
Yeah, I'm very interested that Quebec, when added up there in some provincial parks with all those different methods, because that is another way to do it. You hit it if you've got the resources, funding, and permits and all that. You hit an invasive with everything. Manual, mechanical, chemical, whatever it takes. But I'm not an expert in this at all. But you need different kinds of chemicals to take down like Eurasian milfoil than you do Starry Stonewort, because one's a vascular plant and one is an algae, a macroalgae. So you're talking about herbicides and algicides, and maybe there's some cross uses there, but I wouldn't know. You'd have to talk to somebody specialist about that. But you are talking about two different families of plant. So it's, and again.
Okay, thanks, David.
I'm sorry?
Okay, thanks, David. That was my kick at the can to just propose an alternative there, and whether you had heard of anybody else using any alternative methods like that with any success.
Yeah, no positive success, but it's happening. There's a lot going on out there. So, and there is a local place to me that was talking about doing it, but I don't know if they ever did. I don't know if they ran out of funding or what. But thank you. That matting is it's certainly a viable option. Thanks for the information.
I wonder if, Doug, are you going to tune into the next webinar? It's Dan Larkin from Minnesota is doing that, and he's talking about management things as well. You may be interested in asking that with him, or? Yeah,
I'm going to contact, I believe it's Wildfire Golf Course I'm talking about, isn't it?
Yeah.
Yes.
Isn't that the golf course on Stony Lake?
Yeah.
Yeah.
I'm going to contact them and see if they have a comment as to how well their campaign went last year.
Sure. And could you let me know what they...
Yeah, I will. We actually need to get some feedback from people that spend thousands and try to make an attempt to control weeds. It'd be kind of interesting to see if they were successful or not.
There's another thing I'll add too. There's a scale, what I call a scale factor. I talked to a researcher from this young guy, just went out of my head with college down in Jersey. You know, they took some very small lakes or ponds, and they hit it hard with algicides and stuff with some positive results. But they hit it real hard with algicides season after season. I said, what about a big lake? And he says, nobody knows yet. So scaling these up from like, I don't want to be gloom and doom here, but if they have success at a pond and at a golf course, that's fantastic, right? And that's good information. But trying to scale that up to a cove and a bigger lake, it may or may not work. And that's another part of the research. Whatever method you're trying is how will that work? Scaling up the larger lakes and things. Just throwing that out there.
Yeah, David, I think
Doug,
the method that I'm suggesting. I believe Patty was trying to speak there, so I'll pause.
Before you got off the mats, I was just going to say that a couple years ago, Doug Coleman from Big Cedar came out and looked at my bay because he'd been using mats for several years with Rick Sager. I'm not sure whether it was out of Trent or Fleming at the time. And he said to me they were spending $25,000, $30,000 a year on these mats. And he looked at my situation where I'd just been hand pulling and sort of doing things on my own, and he couldn't believe what my bay looked like compared to his areas that they'd been trying. So he wasn't as keen on the mats as when they had initially started. So you might want to give him a call. He's with Kawartha Lakes Stewards. He's the guy for Big Cedar. So he's been using mats for several years over there, but for Eurasian milk, right? So,
Patty, that's actually Doug. That's on the line.
Patty, that's me you're talking to.
I thought this was Doug Wellman. Sorry.
No.
No.
Oh, I'm sorry. Okay,
This is Doug Palmer. I'm logged.
Okay, so I'm just telling you what you already know.
Yeah.
Sorry about that.
Sorry, I'm logged in as Pixel. I don't know how my phone connected me as Pixel, but my phone is a Pixel phone. So.
Doug's a little. Yeah. Okay.
Yeah.
Well, there you go.
I still think your bay has merit for the possibility of matting, Patty, if you want the truth.
Yeah, okay.
Anyway,
well,
moving on.
It's all covered. Yeah.
Okay,
yeah.
It's after 12, so just wondering if you could let David move along.
I'm fine, whatever you guys want. Get more questions, it's okay. I'm good. I don't know if anybody else.
I'm not sure who's still left. David, could you just do an update and let me know who's still on the line, just so I know who I'm talking to?
Carol, myself, Ann Ambler, Patty McDonald, and Greg Finley. And you.
Okay. Yep, sounds good. All right, thanks very much, folks.
Okay,
Yeah,
thanks.
thank you.
Take care.
Bye,
Bye-bye.
Doug.
Ann, do you have any
Bye-bye.
questions? Okay. All right, folks.
Well, thank you so much, David. It was great.
Well, thank you. This is good.
Have a good one.
That's what it's all about, sharing information. So.
Yeah.
Okay, thanks.